In this video, we're going to be taking a look at microscopes and the various types of questions that you may be asked. Here we see a virtual microscope. Under the microscope, we see a monocot root. This monocot root is viewed at 60 times magnification. What we're going to be asked is, what is the actual size of a monocot root? To figure this out, we need to use the field of view. The field of view for a 60 times magnification is 2400 micrometers, and that's given right in this window here. So that means that the distance across the field of view is 2400 micrometers. We can see this monocot root here takes about half the field of view. Therefore, we know that the monocot root must be right around 1200 micrometers. So that's how you determine the size of an object under magnification, and it can be different for different types of magnification. Your field of view will change. We can see now that two monocot roots could fit inside the field of view. Let's take a look at this question. It says, given the diameter of an average frog blood cell is 0.025 millimeters, how many such cells can be lined up across the diameter of the virtual microscope at 600 times magnification? Well, we're given a few things in the question. We know the field of view is 250 micrometers, and the width of each frog blood cell is 0.025 millimeters. But where did I get this 250 micrometers right here? Well, I got it directly from the microscope. The field of view at 600 times magnification is 250 micrometers. Now we're trying to figure out how many cells can be lined up. To do this, we first need to convert this 0.025 millimeters into micrometers. And how we do that is multiplying by, in 1 millimeter, there's 1,000 micrometers. So multiplying these values together, our millimeters are going to cancel, and we're going to be left with micrometers. So 0.025 millimeters is 25 micrometers. Then we just need to figure out how many cells can be lined up. We can figure that out using the equation here. So the number of cells that can be lined up is equal to the field of view divided by the width of one cell. We know the field of view is 250 micrometers, and the width of one cell is 25 micrometers. Dividing those numbers, we'll get 10 cells. And if we look at our microscope, we could actually line up 10 of these frog blood cells. So that's how you figure out how many cells or how many whatever you're looking at can be fit into the field of view. In this question, an artist has painted a red blood cell to scale. The width of the cell in the painting is 30 centimeters. So we're given 30 centimeters, that's the width of this cell, and we know the scale of the painting is 1 centimeter equals 0.25 micrometers. So we're being asked, what is the actual size of the red blood cell in micrometers? To do this, we know from the scale that 1 centimeter is equal to 0.25 micrometers. Since the cell is 30 centimeters wide in the painting, we know that 30 centimeters multiplied by 1 centimeter, there's 0.25 micrometers in the painting. That means our painting represents a red blood cell that is 7.5 micrometers long. So in this, we've used the scale, centimeters canceled with the centimeters, and we use this scale ratio right here. 1 centimeter is 0.25 micrometers, and that was given right here. So the actual cell is 7.5 micrometers. Now what if we wanted to figure out what the magnification of the painting is? Well, we'd have to use our formula. We know that the magnification is equal to the size of a drawing divided by the object's actual size. The key thing to remember is that both of these values must be in micrometers, or they must be in the same units. So we have to figure out if our blood cell has been drawn or painted at 30 centimeters, how many micrometers is that? Well, we're going to have to use another conversion factor. So the size of the drawing is 30 centimeters, and we know that in 1 centimeter there are 10,000 micrometers. Multiplying these values together, the centimeters is going to cancel with the centimeters, and we're going to be left with 30,000 micrometers. But that's not our answer. That's just the size of the drawing, the numerator in our formula. The object's actual size we figured out in the previous question was 7.5 micrometers. So if we divide those two values together, we'll get 4,000 times. So 30,000 micrometers divided by 7.5 micrometers is 4,000. So our painting is actually 4,000 times bigger than it is in real life. I hope this video has been helpful.